Thank you for coming today to Speeding Up Genomic Discoveries Through Innovation, New Fast Library Quant NGS, and High Fidelity and Long Range Amplification. It's a bit of a mouthful. I'll do my best with it of trying to interconnect seemingly disparate products for you. How many here are familiar with Quantabio? Most good number know us. And I think most are probably familiar with some of the backgrounds. I want to give a quick introduction to us and then kind of hit some of the new product highlights, products that have just released onto the market, which is the Fast Library Quantification Kit. And all that is driven on the Q cycler that I introduced at ASHG last year. So this is sort of a follow-on to, continuing series of talks. And then sort of shift focus from library quantification to some core tools that actually can be used largely for template prep now. Some of them seemingly kind of old technologies, but with, we think, innovative new twists to them that can push things into this current generation of needs for emerging technologies. So just quick background, I was actually one of the founders of Quantum Biosciences. We came out of Life Technologies. I was with them for over 20 years, was on the teams that led a lot of the superscript development, Platinum TAC, and so forth. And so our obvious focus when we came out and we formed our own company was focusing on qPCR, PCR, cDNA synthesis. We did that through engineering really application-specific enzymes, linking those with optimized reagent systems, providing superior application-specific products. Most notable has really been, I think, the tough mix line in enabling amplification and accurate quantification from samples with challenging sample matrices. Here of late, we've been expanding the breadth of the line to extend into the NGS field. A lot of this arose out of our move from Gaithersburg. We relocated in 2016 to Beverly, Massachusetts, and implemented manufacturing and R&D under ISO 13485 standards. And the focus of the teams there at Beverly, largely around enzyme development and workflow development for NGS, nucleic amplification, synthetic biology, and also some gene editing work that we've been playing with. The Spark NGS portfolio has been growing rapidly. We started with a streamlined workflow of enzymatic fragmentation, ligation, or DA tailing, ligation, and amplification those are all the three products that go into that workflow. Recently, we added kind of a very high-performing, really the equivalent of Ampere beads with the Spark Pure Mag. And now recently we've updated the whole library quantification system, and that's what I want to spend the first part of this talk and address for you. So most typical, there are new workflows out there that eliminate the need for library quantification, but the classic NGS workflows for Illumina-based platforms do have a fairly high requirement for library quantification at the end of the process. And that's obviously, why is that important? Really what you want to do is you want to maximize occupancy of your flow cell to get high quality read data. qPCR has really been the gold standard for that process because it quantifies only library molecules in which the P5 and P7 adapter molecules have been attached to either end. And eliminates some off-target things that may happen during library prep. That barriers to using qPCR routinely, is cost. I'd say that the time, complexity of the setup, need to dilute your library samples, that probably doesn't go away. That's consistent with most methods you're going to use. But there can be then inherent technical errors introduced, and really pipetting errors contribute more to inaccuracy in the library measurement, I think, than the qPCR process itself. But we've looked to streamline all of that with our new SparkFast library quant kit. This is sort of just a quick overview of the workflow. I'll go through each step individually, but the net result of this is really looking at faster time to results, improving your lab throughput, at least 60% shorter run times than typical cycling protocols, providing very accurate, reliable quantification over a broad range of GC content and library fragment sizes. Because this is running on the queue, I don't really have time to get into really great details on this. I'll highlight some of this, but this little instrument actually gives you the ability to do very accurate and reliable, highly, quantification down to 1.2-fold differences. What makes this very easy to use, I think, from the start is really it's all in the SparkFast library mix. So this is it's a dye-based master mix. We've made this at a 1.25x concentration, and we've integrated both the P5 and P7 specific primers into the product. So your pipetting setup is very simple. It's just 2 steps. Add master mix to the tube, and then sample. And eventually, we hope to actually have the master mix in the tubes themselves for this, so that all you would have to do is add your sample. It's kind of a next evolution of this. But it's effective for both either 10 microliter or 20 microliter reactions that are typically used. Again, there's a stringent monoclonal antibody hot start used for the TAC in the product. Allows convenient room temperature setup. We've taken this through 20 freeze-thaw cycles. I think subjected it to well over a month at room temperature, and we've seen no degradation of performance. So the master mix keeps things off, and these primers are stable in this mix for a good long time. So this is an example of, this is where we see the performance gains in shortening the run times. Some of this is coming from the SparkFast library master mix, but a good part of it's coming from the Q cycler itself. It's a little bit of a different protocol that we're using, and we found this to translate more uniformly, the 65-degree annealing step, annealing extension step, allowing us to get more accurate capture, broader sequence diversity. So looking at either AT-rich or GC-rich sequences, we get comparable efficiencies with libraries of either source. And on the far side is just the traditional protocol used by the industry standard kit for library, qPCR library quantification. So beyond setting things up, then you're into more of a classic mode of absolute quantification, as absolute as any absolute can be with qPCR. There are lots of caveats around that, of course. But we've tried to simplify this as much as possible and make it as reliable. So we have a set of 6 pre-diluted standards that are now at the conventional 20 picomolar down to 0.00002 picomolar that are provided with basically the industry-leading kit. So your workflow, your familiarity with the process doesn't change, okay? We've gone through great measures to make sure that we have very rigorous quality control around that, so that we get precise CT calls from lot to lot. And these dilutions, these standards have very high linear correlation coefficients. So by simply running the standards on the machine and then your diluted library, you then use linear regression analysis, look at your average CQ values, and you can plug those into an Excel template, and it can kick out basically your size-adjusted, size-normalized library quantification for each library for you. Now, one of the things that's really quite remarkable that enables it, I mean, this is only, this product isn't intended for super high throughput labs, because the machine does only have 48 sites on it, okay? And so if you were running your standards time and time again, it doesn't leave a lot of room for a lot of libraries. But the uniformity and reproducibility of this machine is sufficient that you can merge data into what's called a project file. And so you can either, if you want to run just a single standard as a calibration, as a calibrator, and even that may not be necessary with the kind of reproducibility of results that we've seen, you can run your standards once, and then at least from a given kit, I would say you can just import data in, and you can quantify, you can off of the same standard curve and not have any kind of loss of quality of data. So just are you guys familiar with this little machine at all? So this is like your magnetic cooktop. So this is an aluminum block rotor. It holds the samples, holds the tubes. It runs at about 2,000 RPMs during the qPCR. And when electricity to the magnet's applied, the block heats up. You turn off the electricity and apply fans to it, and it cools down. Because it's a continuous unit of mass, and it's the way the samples are arranged in a circular manner, you have phenomenal site-to-site temperature uniformity from each tube. One of the early things I did with Quanta is we developed an HRM kit, and I did using DNA melting and looked at the temperature differences at sites across what were supposed to be some of the most highly controlled block machines on the market. And you did a heat map, and it varied quite substantially. So if you were looking for a 0.1 degree difference in a class 4 AT SNP, and you had a half a degree variance over the block, it made it a little difficult. This machine greatly simplifies those kind of operations. The rotating nature of it, I think, helps samples equilibrate, or the reaction equilibrate to block temperature quicker. So you have faster ramp rates, you have more uniform ramp rates, and in effect, just better qPCR performance overall. And then this is backed by a very nice data analysis package. It's driven by LinReg algorithms for threshold and CQ calling. So just one example of that. So we're looking at this is a dilution of a pooled bacterial library. So we have 3 different bacteria that we normally use as a model system, and they vary in GC content from about 30% up to 68%. And we mix those together and use those as kind of our standard model system. So this is just the log-fold dilution of that pooled library sample, and we looked at 4 replicate samples over 4 independent runs. And you can see how uniform the data is. Another way to look at that, this is an AT-rich library. AT-rich sequences tend to be, I think, more problematic to amplify because there's greater change slippage during polymerase nascent chain extension. And so this is actually data accumulated from 18 independent runs, a total of 122 different reactions. I think that's actually quite tight over that range. And then if you apply that and actually did the concentration value for this particular dilution of a library, you see that's very tight calls. Again, this is looking at a dilution of our 67% GC library versus a 27% GC library. So over this varying GC content, we have very consistent and constant amplification efficiencies. And so that allows us then to make accurate calls using a single set of standards against a wide range of different NGS libraries. And this just gives an example of sort of that fine level of resolution that can be achieved. We're looking at multiple replicates here with a 1.5-fold difference with each dilution of this particular library. But I guess the real money shot for you here is how if I'm using a Kappa kit right now, how am I going to stack up against an industry standard? So this is we looked at 18 different replicates for each kit here, 5 different RNA-seq libraries that we measured, and overall, I think we have very high concordance between the 2 different kits. So basically, same result, faster time, easier setup, simplified workflow, much less hands time on the bench. The ability to utilize the same set of standards, basically lowering costs for you, and so we're very excited about this, and we hope this will be well embraced by the industry. The feedback has been very positive on it so far. The downside is right now at the moment, the kit is restricted to deployment on the Q. And so the project that we're currently working on is taking the core knowledge that we used in building the kit for the Q and testing it, re-optimizing it. Some of the different machines may require, seem to require slightly different cycling protocols, but we're looking at early next year releasing a universal library kit that then could be deployed on any platform and bring many of those same kind of convenience factors for library quantification to a broader range of qPCR instruments. So I would welcome you to go, if you're interested in our NGS product line. Eleanor was presenting a poster yesterday. I think that's still up. So if you want to look at the data in a little more detail, please visit the poster here. There's trifold brochures available at the booth over at 1620. Take a long walk almost all the way to the posters, and you'll find Quanta kind of buried back there. Or visit our NGS landing page. There's a wealth of resources, and there's a wealth of resources and tutorials and on actually using the SparkFast library quantification kit on the Cube on the website. So to shift gears, so we've looked at quantifying, but then we can look at what can we use to potentially make libraries. And one of those is that one of the trends we're seeing more and more is research is shifting toward looking for longer amplification of longer sequences. A need to multiplex longer sequences, bring those in, looking at different splice variants, things for like using this as template prep for characterizing and doing HLA typing. And so there are a variety of kind of core PCR tools, and some of these have been around for a very long time. So you might ask, why long-range PCR? I mean, this is 26 years old now. I mean, I met Wayne Barnes back when he first did this. It dates me, but some of this technology could be actually older than some of you sitting in the audience right now. But Wayne reported this, and why was it important, and why is it still relevant? I mean, in the old days, people were looking at just trying to clone products, and so they were looking at trying to characterize based on cloning. Now everything's moving into NGS workflows, and whether that is then taking the longer products, fragmenting those, and doing sequencing on Illumina, or using the longer products to feed workflows directly into long-read platforms like PacBio. So there are emerging needs, and I think some of the older technologies that we've been working to improve upon do still have a very firm place as a research tool today still. So what was the revolution behind long-range PCR back in 1993 or so? It's kind of a long time ago. Basically, the enzymes used for high-fidelity PCR, namely these archaeal, polymerases like PFU, vent, deep vent, high fidelity was synonymous with finicky. They were a pain to use. They were not very reliable. They generated low yield. Their processivity of these enzymes is extremely low. They'll add 2 to 4, sometimes 6 nucleotides before they disengage from the template, so they're always hopping back on. And some of that helped contribute to, actually, I think they thought the theory was that to their fidelity. So by monitoring, they could go into a switch mode of base misincorporation, fix it with the 3' exo, and continue nascent chain extension. Other problems with it is that these archaeal polymerases, this was an observation that reported with Roger Laskin back in the mid-90s. We were the first to discover this actually through working on long-range kits, that DU and template DNA the archaeal polymerase would bind very avidly to that. And so that can actually end up being a problem down the line. So I'll explain that in a minute. So the way Wayne got around the problems of yield and specificity with a proofreading polymerase was to basically supplement TAC with a limiting amount of a proofreading polymerase. So it gave you some error correction. Basically, so if you have misincorporation by TAC, you have a abortive nascent chain extension, TAC falls off, proofreader hops on, fixes the base misincorporation, and then TAC can hop back on and have further nascent chain extension. So you get longer products, about usually more in the range of about, I say, 4-fold higher fidelity than TAC alone. What made these products attractive back in that era was really that for anything over a kb, 2 kb and above, you would see much higher yield and more robust performance, kind of broader application to a wider range of templates. But one downside, and I've always done this long and accurate or long PCR to be synonymous with taking a very long time. When I would set up experiments, I mean, if you do the math, right, 20 kb, 20 seconds in nature, and 35 cycles, it's going to take all night. So my productivity, they thought I was working very hard. I would set up reactions at 4:30, throw them in the machine, go home, come back the next morning, run the gel. Goof around during the day, set up another reaction at 4:30, kind of just continue that process. But that lag, really there's a huge delay in your time to result and analysis with that. Also, this prolonged exposure of DNA or your nucleotide pool to high temperature can result in this deamination of cytosine to uracil. When you have uracil and the uracil gets incorporated into the DNA, then you have termination of extension. Now, the team at Strategene had a novel trick of throwing in a DUTPase into the long PCR mix or with their PFU enzymes, and that tended to help, but these long templates would still be subject to deamination, and if a DU did occur, one, the proofreading polymerase would bind to it very avidly. That acts like a sponge. You deplete the polymerase from the reaction, and it also is a potent block to any further nascent chain extension. That's something that Roger Laskin and I showed in our JBC paper back in the mid-90s. So what we've done is we've kind of flipped this on its edge. We've blended TAC. It's actually a slightly precessive TAC variant with a modified proofreading polymerase. This polymerase has very high processivity, and we've mutated the uracil binding site and the read-ahead function and the amine terminus of the enzyme. So it's basically what we would call DU literate. We've provided stringent hot start for every polymerase activity in the mix, so TAC is inactivated at multiple epitopes with a cocktail of monoclonal antibodies, and we have antibodies sequestering both polymerase activity and the 3' exo activity for the proofreading polymerase. So you have a very stable mix that you can set up at room temperature and not have any problems. One thing that was surprising that came out of this mix, and some of this is from this novel proofreading enzyme in combination with the TAC, but also the buffer system utilized in it. But we saw much higher accuracy and fidelity with this particular mix compared to other long-range mixes. In fact, the fidelity measurements that we got in a typical blue-white screen kind of fidelity assay was actually very close to what we saw for enzymes like KOD or PFU alone. So we were seeing, faster extension times. We were seeing much higher yield, much more robust performance over a broader range of templates, all out of this nice, stable 4x concentrated master mix. And one of the surprises from it, too, is that we've been able to multiplex with it quite nicely and do longer templates. So we can look at, again, that came out of a project where we were looking at trying to multiplex multiple HLA loci all at one time. So going into a little bit of just the data, it's not extensive. There are a few primer sets out there that are widely used that everyone looks at. There's an 8.5 kb locus of beta globin, a 17.5 kb locus from beta globin, and a 24 kb fragment of the TPA gene. So we're looking at long range against two of the other major competitors with some of the better performing long range master mixes on the market, and you can see much higher yield at lower input quantities and also faster cycling times with the long-range supermix compared to the other products. And again, these are also 4 other comparisons against 4 other products on the market. And you see the 8.5 kb, the improved yield, and amplification of the 24 kb fragment from relatively low levels of input genomic DNA, actually, and very strong, very specific yield. So I've been very impressed with that. The other advantage of the ISO 1345 manufacturing system is it drives you and really looks at trying to produce a consistent product. And so this just kind of shows the lot-to-lot consistency and also some of the reproducibility for amplifying some of these, more challenging, longer length products with this particular mix. And again, one of the other benefits that we saw from this was we didn't need, with previous versions of this, there was always a need to include something like beta-ene or some kind of a GC-rich enhancer, and we haven't seen that with this particular product so far. Doesn't mean it, some samples may still need that, but overall, just right out of the box and as it's used, it works very, very well. So this just shows multiplexing of fragments ranging from 1 kb to 6 kb from the BRCA gene. The 6-plex here is shown in the panel over on my left, your right. So again, very flexible, very robust performance with this particular master mix. And so we can't reveal who this was with, but we're actively putting this out in different beta tests and different applications testing in different labs, and we always welcome collaborations. So we had a particular client that was interested in higher PCR product yield. They were looking at generating basically targeted panels that were feeding their long-read, PacBio system. This is the gel data that they provided. You do see some higher band intensity on some of these. Overall, what they reported back to us was they felt they saw improved product yield in faster turnaround time, which improved their workflow. And most importantly, because they were getting higher specific product, they were seeing better quality sequence data on their system. So after that, I guess, rather than asking why long range, kind of like why not long range? Seems to be a very versatile system. It has can have very diverse applications, and we think we've done a good job of capturing these different performance attributes and benefits into the product that we're releasing, and we're very excited about it. That said, the other question to ask is, well, what if? Or why can't I have a pure proofreading polymerase with extreme fidelity and capture many of those same properties that we have tried to exhibited with the long-range master mix? And that's what we're looking to achieve with the replica hi-fi tough mix. I don't believe in universal solutions necessarily, and I think these two products can actually sit side by side, and in some applications, one may have superior performance versus the other, and depends on your individual needs. But this has been reported back to us by the labs that have been testing the product as really being a game changer in terms of true high fidelity polymerase. And again, this gets back to looking at eliminating that DU binding in the polymerase, and we've combined this with other processivity enhancing factors. So this particular polymerase is extremely processive, has very fast, it's very robust, it's and inhibitor resistant and able to amplify from crude samples. So if you look at sequencing data, fidelity is one of these things where depending on how you measure it, you can get widely differing results. But even in like that LAC blue-white screening assay, we still see superior fidelity over enzymes, say like fusion or PFU. In sequencing data, we're seeing about a 90-fold lower error rate than TAC. Might be more like 40-fold in looking at that blue-white screen. We have been able to capture fragments as long as 24 kb. You'll see some of the data, and if you were definitely going for length, I think the long range is probably a better option, but we have done very long amplification. Again, we have hot start for both the exo and the polymerase. Proofreading polymerase, 3' exo, you're going to get blunt ends, and this is compatible with DU-containing DNA, which does open the door for its use in applications with either UDG carryover prevention or methylation analysis, where you're looking at bisulfite-treated DNA and you actually do have DU content and you want to amplify that with high fidelity. So overall, we see this to be about 2 to 3 times faster than some of the best SSO7D modified polymerases on the market, generating higher yield, specificity, efficiency. Efficiency was measured in kind of an odd way. It was compatible. You could spike Ava Green actually into the product and use it on a real-time machine. And it's tolerant over a wide range of inhibitors. About the only inhibitor we've seen it really have high sensitivity to has been heparin, but it's withstood, crude blood or whole blood, hematin, humic acid, xylin from, say, complex plant sample matrices, and most of your other kind of classic sort of PCR inhibitor panel that you might run this enzyme against. That's kind of summarized in this panel. I mean, overall, it tends to stand out pretty high. I think, like I said, in terms of absolute fidelity, we've seen very, very good performance out of QFi and SuperFi, nudging out the fidelity of this. That said, it still does have very, very good fidelity that exceeds that of an enzyme like Fusion. But it overachieves in almost all other metrics that you may want to throw against it. So one way to look at fidelity is going to be a matter of accumulated errors and how many cycles you're running. If you're getting more product and less cycles, then you may actually be accumulating fewer errors. There was a very nice paper put out by the scientists at NEB looking at error rates in NGS sequencing, and one of the most common errors that everyone is overlooking is actually this spontaneous deamination of cytosine to uracil. So you're having a C to T transversion in a lot of templates. By running this faster, minimizing cycle time, you're lowering your susceptibility to those sorts of DNA damage. This just sort of shows the recommended cycling times provided by the different products on the market. I mean, with some things it could be very fast. Under 1 kb, you can get away with like a 1 second extension time, just bouncing it up and down. Mostly, I think it finds its sweet spot really in kind of this 1 to 10 kb range. Over 10 kb remains to be seen, really. I mean, like I said, we've done 24 kb. How reliable that is, we'll see. So, again, just this on pure DNA on a Lambda template. I say we've seen comparable performance, 10 kb to Q5. With the 20 kb achieving slightly higher yield, and in both cases doing that in faster cycling times. It's a bit of a messy data. I mean, you can clearly see that if you're looking at smaller fragments from complex genomic DNA, that the enzyme's performing exceptionally well. Longer fragments, now these were, if you remember the long PCR data, these 2 fragments were extremely robust and extremely specific with the long range. Probably going to need a little more optimization around with this high fidelity enzyme, but we did still manage to capture the 24 kb fragment, albeit with considerably lower yield. I think more important to look at is more common size fragments. We see it perform well and outperform these other, what I would consider to be state-of-the-art high fidelity polymerases on a wide range of kind of normal targets of varying GC content. So this again was a feedback from, again, an outside tester. He had done an annealing gradient to see where he would get best results. So overall, he was very happy, and it was outperforming his current status quo enzyme and lowering his time to actually proceeding with his protein expression experiments. One of the more interesting ones is we had a client look at this to feed library amplification for their PacBio system. And so if we look at, they had been using Kappa Hi-Fi with this is their kind of status quo, and this shows the bioanalyzer of the final amplified library. And so with a 5-second extension time using the the LR-fast, this is the high fidelity, the hi-fi tough mix. They were actually achieving greater product yield, broader fragment size ranges, a longer mode sequence frequency here, and more defined product. And so they're very encouraged by this, and this is one of the things that we're really looking to expand down the road with this. I was talking with another gentleman at a booth, and he inspired me to get back at the bench and take a look at Fragile X and look at how this polymerase can read through repeats. One of the problems with repeats is you have breathing of DNA during PCR, and you can have misindexing. And usually, I've seen more processive polymerases actually more faithfully capture, repeat regions. And since this is a very processive polymerase, I suspect it will work exceptionally well in that particular application area. So, just in summary. So replica hi-fi tough mix, we feel it's demonstrated superior performance compared to other state-of-the-art, polymerases. I think the real proof in the pudding is when you put this in people's hands and they're asking, when can I buy this? So that's one thing that makes you feel very good about the job that you've done with the product. Where we're looking to go with this down the road is we're hoping to get this out the door early Q1 next year. Personally, I want to look at more challenging applications, get a better, clearer design around primer design requirements, TMs, and how those marry to specific cycling protocols to improve your probability of success and your success rate when you take the product and you use it. Beyond that, we're looking to expand the breadth of applications, and we welcome collaborations in a variety of areas. We have a number of very novel thermostable reverse transcriptases in our development pipeline, so we think this could feed very nicely into full-length RNA-seq workflows on long-read platforms when we combine these two products. Because it has the capability of copying DU-containing DNA, it's obviously an ideal candidate to throw at methylation studies where you're doing using bisulfite-treated DNA. And we feel that it can also serve as both for a generic and targeted template prep from crude samples, again feeding either conventional Illumina NGS workflows or emerging long-read workflows. So with that, I'd like to thank my R&D team at Beverly, and also a note of high thanks to the marketing team there. Nothing happens without them actually helping me drive this and pull things together. And so both Moniel and Eleanor are here in the back if you have questions about anything. If you want more information on the Q, I do have last year's talk on my computer and be happy to go through some of the benefits of the Q with you over at the booth as well. So with that, thank you, and open the floor to any questions you might have.
PCR is that you can end up with a lot of hybrid reads. So the enzyme falls off halfway, so to speak, and that is then used as a primer on the other allele. Do you have a solution for that, or is your tough mix any better than other products in that regard?
Well, I think, yeah, the question relates to sort of chimera formation during long-range amplification. I mean, so some of the longer extension times, there's ample opportunity for, especially if you're amplifying multiple alleles, for those two products where there's homology, high homology to index. We haven't looked at it that closely. Like I said, we do have clients that are looking at this for HLA typing. I don't know how tightly, the overlap, homologous those various sequences are. But that's a very interesting question to study in greater depth. To date, we've just looked at, trying to achieve specific amplification. I think the proof will come in sort of looking at panels and targeted panels and seeing what comes out of the sequence data with these enzymes.
The long range, the AccuStart long range supermix?
Yeah.
How fast was it?
So for multiplexing, anytime you start multiplexing, I think you need to give the enzyme a little more time. and probably go back to more around that minute per KB range. For individual targets, like the 17 and a half KB, I think we were doing 15 to 20 seconds per KB with that.
15 to 20 seconds.
So that's lowering it substantially from a minute plus, and typically back in the old days, dating myself a little, as you usually we would run 20, 25 cycles, and then we would auto increment extension times in order to get amplification of long products. So far, we haven't seen any need for that with this master mix. It seems to rip through and copy everything to completion. So.